In this lesson, we're going to talk about Cheminformatics and QSAR, which is a type of ligand-based drug discovery. So this is the objectives of this presentation, and we're going to be covering the following topics on Cheminformatics and QSAR, which includes common questions that are asked in the field. What is Cheminformatics, QSAR? Use cases of Cheminformatics and QSAR, typical workflow of Cheminformatics and QSAR, as well as the computational tools that are used for Cheminformatics and QSAR. So you can see here that I've color-coded the questions that are commonly asked. And so here, I've color-coded the common questions that are asked in the field of Cheminformatics and QSAR. And so the green color are related to structure-based drug discovery, and the blue color are related to ligand-based drug discovery. So let's read a few of this as an example. So some examples of common questions pertaining to ligand-based drug discovery includes how similar are two compounds, A and B? And then the next one would be, if I have a query compound, then I would like the computer to search for similar compounds to my query compound that are at a similarity level of 70%. Or another common question would be, what is the chemical diversity of my compound library? Or how about, what is the low energy conformation or the three-dimensional spatial orientation of the compound that I'm interested in? Or how about, given the experimentally determined bioactivity data, which scaffold or functional groups contributed favorably or unfavorably to the biological activity of interest? And some common questions for the structure-based drug discovery would be pertaining to drug repositioning. So from a set of known FDA-approved drugs, which one can inhibit my target protein of interest? So remember that FDA-approved drugs are drugs that have been approved to treat a particular disease. And by performing repositioning, it means that we're simply finding a new or novel therapeutic indication for an existing drug. So jokingly, a lot of people likes to use the term teaching an old drug a new trick, because a drug that has originally been proposed to treat disease A can now be used to treat disease B. And the reason for this is that in any experimentation, it is impossible to test all of the theoretically possible disease that a drug can treat, because there are more than 30,000 proteins, and it would simply break the bank to test a single compound to all 30,000 proteins. And so in any typical projects, compounds could be tested against a couple hundreds, if lucky, or even thousands of proteins. And so as you can see, there's a lot of empty spaces in between, and therefore computational approaches will be useful for filling in the blank. Or how about, given another example, which is related to the structure-based, is given an orphan receptor, are there any potential ligands for it? So an orphan receptor is a receptor that is currently have no report that a ligand can bind to it. And therefore, we could use the concept of protein similarity in order to find ligands or drugs that are able to inhibit one protein. Let's say it inhibits protein A, and because protein B is very similar to protein A, therefore it is hypothesized that there's a possibility that compounds that could inhibit protein A would also have activity against protein B. And therefore, ligands that are known to bind to an existing receptor could also bind to an orphan receptor as well. And actually, this is the basis for drug repositioning. Okay, so let's take an example of the first common questions that was asked. So how similar are molecules 1 and 2? So you could simply take a look at the chemical structure, and then you could tell the difference. That mostly they're similar, as indicated by the black color here. And they're different in the two locations here, as indicated by the red color. And so you can see that at the green position, there's no CH3 here. But here, in molecule 2, there is a CH3. And vice versa. For the red color, there's CH3 in molecule 1 at this position, but it's absent in molecule 2. So another common question would be, retrieve all known compounds that are 70% similar to my query compound. So to the left here, this is a compound, or the query compound, that we have drawn into a database called Chembo. And so Chembo has this functionality in which you could adjust the similarity level threshold. So here we specify to be 70%. And then upon searching for it, we get a list of results, meaning that compounds that are 70% similar to the query compound will be displayed here to the right. And so this will be useful if you want to see what is the possible bioactivity for your query compound of interest, even before you perform your experimental testing. Another common question would be, given the experimentally determined bioactivity data, which could come from original research articles, or it could come from your own experimental research, which scaffolds or functional groups contribute favorably or unfavorably to the biological activity of interest. So what this means is that we would like to see which fragments of the molecule that are present in the ligand will contribute to it being active or will contribute to it being inactive. And so such knowledge will be useful for medicinal chemists in the design of more robust drugs. And so common questions that are related towards the structure-based drug discovery, as I have mentioned already, would be, given a library of existing compound with known bioactivity, what will be the likely activity of my query compound? Or how about, given an orphan receptor, are there any potential ligands for it? Or drug repositioning, if you have known FDA-approved drugs, which one can inhibit the protein of interest? And so all of this could be answered or summarized by this schematic summary. And so this one was created by Cortez Siriano et al. And published in MedChem Communications. And so what the illustration here shows you is that in this top left quadrant, this is the known bioactivity data that you have. And so in the rows, you have A, B, C through G, which are the target protein space. And in the columns 1 through 7, you have the ligand space or the drugs. And so the intersection between the target and the ligand is the tested activity of a particular ligand toward a particular protein. And so in the blue color here, this shows a cross-section of what QSAR models can do. And so QSAR model will essentially take in a collection of ligands, shown here 1 through 7, that have been experimentally tested against only one target protein. And here it is shown as A. And so if you would like to build QSAR models for all of the data here, you're going to have to build separate independent models. One for A, one for B, one for C until G. And each model will comprise of the bioactivity data for all 7 ligands. Or another would be in bioinformatics, you could investigate the interactions between each of the 7 proteins here against a single ligand. So typically you would apply molecular docking to do that, where you would like to calculate the docking energy of a particular ligand to the target protein. Or another could be you could perform a protein QSAR as well. And there is a technology called proteochemometric that will allow you to combine all information here, all target proteins, all ligands, into one model. And so that is called proteochemometric. And the great thing about modeling here is that it will allow you to extrapolate the activity of an unknown compound. And so here, for example, if you have already trained your model based on these information in the top left for the target and ligand space, let's say that you have a new target, and the new target belongs to the protein family here, or the sequence resembles that in the target space, then you're able to make predictions on the likely activity of the bioactivity of the 7 ligands against the new target without even having to perform any experimentation. Or you could also do the same here, whereby if you have a new compound, let's say compound 8, then your existing model could also be made to make a prediction on the new compound 8. So it should be noted here that the new compound or the new target could be obtained from the individual models here, or even from the proteochemometric. Or how about if you have a new compound against a new target protein, you could also do that as well. And so that is known as bioactivity extrapolation, and the other two here are known bioactivity interpolation. And so all of these questions are related and can be answered by this schematic summary. So take a moment to explore what I have mentioned, pause the video, and then proceed if you would like to. So all of the explanation has already been made, but actually it's highlighting it. So why don't I start over again to show you one by one. So I have mentioned already that given a library of existing compound analogs with known activity, what will be the likely activity of my query compound? So my query compound is shown here in the new compound. And so the models are built here in QSAR or bioinformatics. And how about here? Given an orphan receptor, are there any potential ligands for it? So let's say that the new target is an orphan receptor, and we already have some existing information about the other related targets. So let's say that the new target is related to the target here. And therefore, we know the activities of each target against each of the ligands. Therefore, in this particular prediction model, it will be able to tell us for a orphan receptor, which is a new target, what is the likely bioactivity data. And so it will be able to make a prediction. And so such model that could be doing this is is proteochemometric models. Or how about here? We already have some models that we have built, and we have a new compound, which could be a FDA-approved drug. So based on the existing model that we have, we could predict the activity of the FDA-approved drug against the targets that we have investigated. Or how about here? Let's say that we already have built some models, and then we would like to know for our compounds of interest, spanning the columns here, which potential target will it be able to inhibit? And so the models shown in the gray color could be used to make a predictions here. Or how about here? Given the models built in the gray color here, can buy what type of compounds can bind and modulate the bioactivity of the target protein of interest, shown here to the left. And another common question that is asked is, from a set of compound analogs, what is the most important functional group or the most important molecular fragments? Let's say that if we take a molecule and we break it down to the fragments, we want to know which fragment is important for the bioactivity. And so shown here is an example from a anti-cycling activity QSAR study from our research group. And so what we did was that we calculated the molecular fingerprints of the molecules that have anti-cycling activity and those that have weak or no anti-cycling activity. And then we built a random forest model, and then we investigated and discovered the top performing features that are shown here to the left, as indicated by the Gini index, which comes from the random forest model. And as for each of these top descriptors, we took an in-depth analysis of it by using box plot analysis. And what we did was that we stratified the compound data according to the activity class of active and inactive. And what we wanted to do was investigate for the active class and the inactive class, which particular molecular fingerprint has more of each. Okay, and so those that have the 3 asterisk are known to have statistically significant difference in the numbers of compounds having this particular substructure in the active or in the inactive class label. Another common question would be, how extensive are my compound of interest in terms of their chemical diversity? So this is also taken from one of our research group's publication from Dr. Nuttapat Anuwongcharoen. And the study investigated the origins of the bioactivity of inhibitors that could bind to influenza A and B neuraminidase. And so they're color-coded according to their bioactivity, as shown in green for actives, red for inactives. And so to the left here, it shows the scores plot, which shows the chemical diversity of the compounds. And so we could see a distinct classification or clustering here between the two classes. And then based on the scores plot here, we superimposed it with the loadings plot, as shown by the labels of the loadings factor or the molecular descriptors that are investigated. And so the color here are shown according to the scores plot, which are superimposed here together with the loadings plot. And then we can see which region, as indicated by green, are occupied by the descriptors. So what this will essentially tell us is that the descriptors falling inside the colored regions are important for the particular bioactivity. Therefore, for example, LUMO, RBN, HOMO-LUMO, number of hydrogen bond acceptor, TPSA, number of hydrogen bond donor, are shown to be inside the green region, and therefore they are important for the active class. On the other hand, energy, number of cyclic rings, HOMO, energy, highest occupied molecular orbital, and the A log P are found to be occupied in the inactive regions, and therefore they're important for the inactive compound. And then the bottom part is shown for the influenza B inhibitors. Like for example, HOMO and energy descriptors are found to be in the inactive group for both influenza A and B. And so from this PCA analysis, we are able to look at the chemical diversity in relation to the bioactivity space as well. So another common question is, given an existing molecule, how can I generate a novel molecule? And so let's say that you have the original compound shown here, and you want to modify the molecular fragments indicated by the green color here to the red color here by replacing the methyl group with the chlorine. You can see that the original activity is inactive, but upon modification, it becomes active. And so such substructure substitution is typically done in medicinal chemistry. And as you can see here, it gives rise to active compounds by some replacement of the functional groups. So analyzing the structure-activity relationship will allow you to pinpoint the important positions in the molecular structure of the ligand. So this is a surface plot of the conformational space of the molecules shown here. And molecules are very dynamic and they have several degrees of freedom because the single bonds are able to rotate. And therefore, this will give rise to several possible molecular conformation. And so a conformation is a three-dimensional arrangement of atoms in a molecule. And because molecules have atoms connected by bonds, therefore the rotation of the bonds or atoms will give rise to different dihedral angles. And so this correspondingly will give rise to different conformation of the molecule. And each conformation will have different molecular energy. And therefore, in this plot, you could see that upon rotation of the dihedral angles, we could see the differing in the energy shown in the color region here. And another common investigation that is typically done in cheminformatics and QSAR is to study the ATMET properties of the compound. And so the ATMET properties determine the pharmacological properties of the ligands when it is inside the human body. And so collectively, we call this the pharmacokinetic properties. And they include the absorption, distribution, metabolism, excretion, toxicity. And so for absorption, a common question would be, will my query compound be absorbed into the body? And so this is mostly determined by the solubility of the molecule. And pertaining to the distribution, one could ask, how will it be distributed in the body? Will it go to be distributed in the extracellular space? Will it be inside the cells? Will it go to the brain if it could pass the blood-brain barrier? Will it localize in the liver? So this really essentially determines also by the absorption as well. And so what determines this is the substructure of the molecule. Another property would be metabolism, and the question would be, what would be the kinetic of its metabolism? And for excretion, how fast could it be excreted? And for toxicity, will the compound be toxic? Okay, so by analyzing the structure-activity relationship of compounds, we could get a more understanding of the ATMET properties. So what is cheminformatics? So advancements in high-throughput screening technology together with large-scale databases, it gives rise to large collection of structural data, and subsequently this also gives rise to the field of cheminformatics. And the structural data pertains to the chemical structure. And so the thing is, when there is more sophisticated robotics and automation, it gives rise to more experiments, more high-throughput experiments, and therefore we get more data about the chemical properties, physical properties, as well as the biological activities of a large collection of libraries of compounds. And therefore, such big data on the chemical libraries and their biological activities give rise to the field known as cheminformatics, because it is essential for informatics to help make sense of the big chemical data. So you can see here in the image, this is a robotic machinery that will perform the bioactivity assay in an automated fashion. So let's take a look here. What is cheminformatics? So the term cheminformatics was coined by Frank Brown, who defines the term to be cheminformatics or chemoinformatics brings together all of the information resources that a scientist needs to optimize the properties of a ligand in order for it to become a drug. So you can see here that it is using data-driven approaches to make sense or connect the dots between a compound ligand and it being a drug. And so you can see here that the intermediate part from a ligand to a drug, there's a lot of information that needs to be taken into consideration in order to make a ligand become a drug. So let's take a look at some of the examples here. So some of the common data storage for compounds would be in a flat file, such as a common separated value, SQLite, or JSON, or it could also be stored in databases such as SQL. And compounds in cheminformatics are commonly represented in the notations of SMILES, SMARTS, and also SLN. And for machine learning purposes, as well as for the calculation of molecular similarity, molecules could be described quantitatively or qualitatively by molecular descriptors and molecular fingerprint. And the molecular similarity could be determined by the Tanimoto coefficient, whereby a coefficient of 0 means that two compounds are totally different, and a value of 1 means that two compounds are exactly the same, so they're identical. And so any value in between will tell you how similar two compounds are. And so if it is approaching 1, then it means that two compounds are highly similar. Substructure search. So when searching a chemical library for the presence of a specific substructure, you could make use of the generated fingerprint and also the Tanimoto coefficient. So that will allow you to find similar substructure and similar compounds. But before doing such search, you would also have to fragment the molecule as well. And so when you take a molecule and you fragment it, you get the substructure. And so it should be noted here that the search here could be for both the molecule as a whole, or it could also be for the substructure itself. And it is very important to note here that molecule data or chemical structure data are presented in hundreds of different file formats. And therefore, there are tools such as OpenBabel that will allow you to interconvert between the different structure formats. Let's now take a look at what is QSAR. So QSAR is Quantitative Structure Activity Relationship, and the acronym is QSAR. And so, in a nutshell, it is an approach for identifying the mathematical correlations between molecular features of compounds with their corresponding biological activity. And so a schematic diagram is shown here, whereby molecules could be described by their molecular fingerprints or molecular descriptors, and then we could fuse together all of the information into a data set. And so here you can see in the columns the various molecular descriptors, X1 through X10, and then the Y column would be the bioactivity class label, where a value of 1 would be compounds are active, values of 0 would be compounds are inactive. And then the data set here will be used for model building, whereby we apply machine learning to learn from the retrospective data, and then the model could be applied to make prediction on a new molecule, which we also calculate the molecular descriptor and use the molecular descriptor as input to the molecule, and the molecule will make a prediction on the bioactivity label. And shown here, we see that the molecule 3 will be predicted to be 1, which means that it is an active compound. And another great thing about the machine learning model that has been built is that we could extract useful knowledge out of that. Particularly, we could extract which important features are there so that we will be able to understand which substructure or which chemical features are important for a molecule being active or being inactive. And so that knowledge will be used by medicinal chemists in their design of a new and robust version of the chemical library. So some of the use cases of QSAR includes it helps to provide important properties. For example, the feature importance sheds light on which substructure are important or which molecular properties are important. It allows you to filter compounds for the desired bioactivity, and the predicted bioactivity will help us in prioritizing compounds, meaning that we're able to rank the compounds in order of importance. And let's say that you have a large collection of libraries of compound, and by applying QSAR, you're able to prioritize and select the top 10 to use for your experimental investigation, because experiments are expensive, and therefore you would like to use a smaller subset that you believe to have potentials in providing robust bioactivity. Otherwise, testing experimentally against the entire library could be infeasible and very expensive. QSAR can also aid in compound modification because the feature importance will indicate which substructure or which molecular fragment are favorable and unfavorable. So here it shows the typical workflow of a Cheminformatic or QSAR study. So you start by dataset compilation, whereby you could collect the data from public databases, from primary literature, original research article. You would combine it, and then you would select a subset of the data for further investigation. And then when you have a data subset, you would perform some cleaning and pre-processing. You would calculate the descriptor, and because the features or the descriptor, there are too many of them, you have to select those that are providing meaningful information, and then you would apply feature selection in order to do that. And when you're doing classification, your classes could be imbalanced, and therefore you could perform data balancing. And then for model building, in order to evaluate the model performance, you would perform data splitting. And then for each of the data split, for the training set, you would use it for constructing a QSAR model or a machine learning model, and then you would evaluate the model performance metrics, and if it is satisfactory, you would use it to identify important features as well as use it to interpret the important features in order to help medicinal chemists and also help chemists in the design of future and more robust compounds. So this illustration here is taken from a recent book chapter that our research group has written. And so this is a high-level schematic illustration that summarizes how the existing resources in cheminformatics and bioinformatics, the various databases and the various experimental approaches, are able to help us design drugs at various levels, from the fragment-based level to the ligand-based level to the structure-based level, and also to the systems-based level. And so you can see here that we apply cheminformatics, bioinformatics, as well as cheminformatics approach, as well as chemogenomics approach. So more information could be provided in this particular article, which explains how to design a successful computational drug discovery project. And so the field of cheminformatics and QSAR, the objective is to find potent drugs. And this endeavor is a multi-objective optimization problem, whereby a drug need not only target the protein of interest, but it must also provide additional properties, such as favorable pharmacokinetic or ADMET properties. So you can see here in this illustration, aside by being able to have potency, for example, to kill cancer cells or microbe cells, it would also have to be safe for humans, as elucidated by the ADMET property. So the balance here is between potency and ADMET property. So the multi-objective optimization would have the following desirable characteristics of a drug. Number one, it binds selectively to the target protein. Number two, it should be absorbed in the stomach for oral drugs. It is able to permeate gut wall or cell wall, or even the blood-brain barrier, so that it could reach the target site. Number four, it is metabolically stable. Number five, it is non-toxic. And most importantly of all, it can be synthesized economically. And number six, and most importantly of all, is it could be synthesized. Because a compound that is very difficult to synthesize, or which requires several steps or too many steps to synthesize, it might pose a challenge for the organic chemist. And so in order to achieve the balance between potency and ADMET properties, the chemical structure will need to be optimized. And so there's a term called Bioisosteric replacement, which is a strategy used by medicinal chemists, and it is instrumental in the modification of chemical structures. So in essence, functional groups will be replaced by chemically similar ones, such that they maintain the same biological activity in binding the target protein, and while at the same time it gives rise to better ADMET properties. So here you can see that molecules shown are quite similar in that they have replacement of functional groups to those that are chemically similar. And the key part here is that when you replace functional groups to a chemically similar one, key challenge is to maintain the same biological activity. And such modifications will have to be made in relation to balancing between the potency and the ADMET property. So this is a very difficult task. So how can you represent molecules in the computer? Well, there are software such as the one provided by ChemExon called Marvin Sketch. And the one on the left, you could represent it in 2D. And there is also a Marvin Space that could allow you to represent it in 3D. And the Avogadro software allows you to represent it in 3D as well. And you could even optimize the molecular structure as well using molecular mechanics. So these softwares are available free for academic license. However, if you're coming from commercial industries, then it will require upfront payment. Avogadro is an open source, so it is free. And so thank you for watching this lesson, and I hope that it was helpful for your understanding of the topic about cheminformatics and QSAR.